Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Assalamu alaikum, dear students. In the previous lecture, we have discussed about C4 cycle or Hatch and slag pathway or dicarboxylic acid cycle. Today we will take up photorespiration. This photorespiration, it is also known as C2 cycle or glyoxylate cycle. This photorespiration, as the name indicates, photo means light and respiration. That means light dependent respiration. Generally respiration is independent, but this is actually a type of respiration which is light dependent. And if we can say it is a light dependent oxidation of intermediates of carbon assimilation, which is accompanied by the absorption of oxygen and release of carbon dioxide. So in respiration, intake of oxygen and uptake of carbon dioxide takes place. Same thing happens here, but it takes place in the presence of light. That is, it is light dependent. It is light dependent oxidation of intermediates of carbon assimilation. That is why we call it as photorespiration. Most of the intermediates of this cycle, they are C2 compounds. They are carbon 2 compounds. That is why we also call it as C2 cycle. And the first product of this cycle is also a C2 compound, that is phosphoglycolate or glycolate. That is why we call it as glycolate cycle, and this is also known as glyoxylate cycle. This is also known as glyoxylate cycle. And this cycle, it was first reported by Dicker and Teo in 1959 in case of tobacco leaves. And it was first discovered by Dicker and Teo in 1959 in tobacco leaves. Subsequently, first photorespiration was found to be universal in C3 plants. It was found to be universal in C3 plants. C4 plants do not show this photorespiration, but C3 plants show this photorespiration, and it is said to be universal among the C3 plants. It involves 3 cell organelles, and this photorespiration, it involves 3 cell organelles, that is chloroplasts, peroxisomes and mitochondria. And it involves 3 cell organelles, chloroplasts, peroxisomes and mitochondria. And it involves these 3 cell organelles. The process of photorespiration occurs in a series of reactions, which is also called as photosynthetic carbon oxidation. And it involves a series of cyclic reactions which are known as photosynthetic carbon oxidation, which involves photosynthetic carbon assimilation, and it involves photosynthetic carbon oxidation. That is why we also called it as PCO cycle. Now this PCO cycle, that is photorespiration, it is the characteristic of C3 plants, and it generally depends upon light, temperature and oxygen. If the concentration of oxygen, temperature and light is high, and if the concentration of oxygen is higher and we have a higher temperature, at that time RUBP, ribulose bisphosphate carboxylase, it loses its affinity towards carbon dioxide and it becomes it gains its affinity towards oxygen. Yani it becomes a ribulose bisphosphate oxygenase and reacts with oxygen. When the temperature is high, light intensity is very high, and oxygen concentration is high, its affinity towards carbon dioxide it gets lost, and it shows its affinity towards oxygen, and it reacts with oxygen and produces a C2 compound. Actually, it produces two compounds, one 2 carbon compound phosphoglycolate and one C3 compound, that is 3 phosphoglyceric acid. 3 phosphoglyceric acid. While as in case of C3 cycle, we get two molecules of 3 phosphoglyceric acid. Here we get only one molecule of 3 phosphoglyceric acid and one molecule of 2 phosphoglycolate. This phosphoglycolate, any RUBP, ribulose bisphosphate, with the help of RUBP oxygenase, it produces one molecule of 2 carbon compound phosphoglycolate and one molecule of 3 phosphoglyceric acid. This phosphoglycolate, it undergoes, with the help of phosphatase, it produces glycolate. And here phosphorus gets released from this phosphoglycolate and it becomes glycolate. And this glycolate then moves out of the chloroplast and enters into the peroxisomes. In the peroxisome, this glycolate, it with the help of enzyme glycolate oxidase, it undergoes oxidation. It undergoes oxidation and produces peroxide and glyoxylic acid. This peroxide with help of catalase produces water and oxygen. And what we can say, with the help of glycolate oxidase, this glycolate produces glyoxylic acid or glyoxylate. This glyoxylate is also a 2 carbon compound. It undergoes transamination. With the help of this ammonia, it produces glycine, which is also a 2 carbon compound. Glycine, it undergoes amination to produce glycine. This glycine enters into the third cell organ, that is mitochondria. Here, 2 molecules of glycine, they react to produce one molecule of serine, which is a 3 carbon compound, and here also one molecule of carbon dioxide gets lost, one ammonia molecule gets produced with the help of enzyme glycine decarboxylase. Here also reducing power, that is NADPH, also gets produced. This carbon dioxide which is produced here, it actually gets lost. Here. Now this serine, 3 carbon serine, it re-enters into the mitochondria, into the peroxisomes. Here it undergoes deamination. And produces hydroxypyruvic acid. This hydroxypyruvic acid then undergoes dehydrogenation and it produces glycerate, which is a 3 carbon compound. This glycerate then enters into the chloroplast, and here it undergoes phosphorylation to produce 3 phosphoglyceric acid. This 3 phosphoglyceric acid enters into the Calvin cycle. To regenerate the RUBP, and this RUBP is then ready to get another molecule of carbon dioxide and oxygen. If the concentration of carbon dioxide is higher, then carbon dioxide reacts with it, and if the concentration of oxygen is higher, then oxygen reacts with it. In this way, this C2 cycle or photorespiration or glyoxylate cycle gets produced. We have seen that in glyoxylate, in photorespiration, no energy is produced. That is why we call it a wasteful process, and it is the characteristic of C3 plants only. This is all about photorespiration. Thank you. Assalamu alaikum wa rahmatullah.